In this protocol, we can identify the interactions between proteins and DNA, revealing the transcriptional regulation of the gene. This helps to understand the molecular mechanisms in the cells. We modify the pre-existing protocols and then establish the straightforward and clearer assay protocols. Users simply follow the protocol from the first step so that the assay can be done successfully.
To prepare cross-linked chromatin, in a fume hood, add 4 ml of 1% VCAP cell culture media and 0.229 ml of 18.5% PFA to a 6 cm dish. Gently swirl the dish to distribute the PFA evenly. Incubate at room temperature for exactly 10 minutes. Then, add 0.47 ml of 1.25 molar glycine solution to the dish and incubate at room temperature for 5 minutes to stop further cross-linking. After washing the cells according to the manuscript, scrape the cells, and transfer the cell suspension to a 1.5 ml microcentrifuge tube. Centrifuge the tube with the cell suspension at 3000 times G for 5 minutes at room temperature to recover the cell pellet. Use a pipette to completely remove the PBS. Record the cell number per tube, and store the cell pellet at -80 degrees Celsius. To perform cell lysis, first thaw the stored VCAP cell pellets on ice. Add 300 microliters of the prepared ChIP cell lysis buffer